All right, so here is where we left off with chapter 4. Now this picture is in the book on page 76. This is the picture of the typical prokaryotic cell. And like I said previously, you're not going to be tested on any pictures or anything like that. But I do like to show the picture of the prokaryotic cell just so you are familiarized with it. So again, the prokaryotic cell is smaller and more simple than the eukaryotic cells are. The DNA is in an area called the nucleoid, and then the ribosomes exist inside the cell as just kind of loose, free ribosomes. And the ribosomes, of course, their function is to make proteins. So then working our way from the inside to the outside, we get to the plasma membrane, which is very similar to the plasma membrane of eukaryotic cells. And then we get to the cell wall. So in this picture, the cell wall is the darker brown color. And we will definitely spend more time talking about the cell wall because it's very, very important in bacteria. Then the outermost layer is called the capsule. Now, not all bacteria have a capsule. Bacteria that contains a capsule, we use the term encapsulated. So there will be more information about that on the next slide. And then, of course, we have the flagella. So in bacteria, the flagella can vary, which we'll have that slide coming up here in a second as well. But of course, the flagella are used in movement or motion. Now, in this picture, they use the term fimbria, and I kind of wish they used the term cilia instead. But we will talk about fimbria and we will talk about the pilus. Again, you're not going to be tested on this picture, but because bacteria or prokaryotic cells are so important to our class, we certainly need to at least look at the picture and run through it here. All right. So hopefully in A&P1, you got the term glycocalyx. So glycocalyx is just kind of that general term for the mixture of proteins and carbohydrates on the outermost layer of a cell. So in bacterial cells, if the glycocalyx is a thick organized layer, that's what the capsule is. So the capsule, if it is present, can give the bacteria certain abilities, such as the ability to escape phagocytosis, which we will talk about later in the semester, or to produce certain dangerous toxins, to attach to a host cell. Basically, the more kind of organized and advanced the capsule is, the potentially more dangerous the bacteria is to a human. And an unorganized glycocalyx is called a slime layer instead of an actual capsule. So again, flagella, singular is one flagellum, and if they have more than one, it's called flagella, and it is for movement or moving around. So for this picture on page 77, you're going to just mostly treat these terms as vocabulary words. So once again, you're not going to be tested on identifying the pictures. You won't have these pictures on the exam, but you do need to know the terms. These terms relate to the various different types of flagella arrangements that can be found in bacteria. So first, we have a peritrichous flagella arrangement, and peritrichous means lots of flagella all over the cell. So as you can see, this bacterial cell has lots and lots of flagella all over the place. It almost looks like it has, a whole bunch of long hair kind of streaming off of it, and that's peritrichous. So monotrichous, so mono means one. So this bacterial cell kind of sort of resembles a sperm cell because it just has one single flagellum. So that is monotrichous. Lophotrichus is a bacterial cell that has several flagella in one single location. So if you notice here, you see the word polar because all of flagella has just one single origin point or attachment point. So Lophotrichus means several flagella in one single point. And then amphitrichus, amphi means both or two. So you can see amphitrichus means flagella at both ends of the cell. So trichus, the word trick, T-R-I-C-H, means hair because flagella is hair-like. It looks like hair on the cell. So that's why all of these terms referring to flagella have the term trichus. So again, just treat these as vocabulary words and you should be good to go. All right, so some more very important, unique bacterial cell terms. Axial filaments are bundles of fibers that allow a certain group of bacteria to move in a corkscrew-like manner. So axial filaments are found on the bacteria that cause Lyme disease and syphilis. So Lyme disease and syphilis are two diseases that we will definitely talk more about later in the semester. But very, very important to know about Lyme disease in particular, because in Delaware, we live in the hot zone of Lyme disease infections. So we have a very, very high rate of Lyme disease in our state. Lyme disease is transmitted by ticks. So I always make sure to tell all of my students that, you know, if you spend a lot of time outdoors, if you have a lot of pets that go in and out, you know, anytime that you're potentially exposed to ticks, you should definitely get tested for Lyme disease every year. And then especially like if you feel very run down, tired, if you feel like, you know, you get kind of sick a lot, you get, you know, like flu-like symptoms a lot, you know, body aches and soreness and just, you know, feeling tired and run down. That can be signs of Lyme disease. It's very important to get tested and treated for Lyme disease because if it us untreated for several years, it can actually cause brain issues. It can actually cause dementia. And again, we live in an area that has very high rates of Lyme disease. But in terms of this topic here, because like I said, we will definitely talk about Lyme disease as well as syphilis. The diseases are very different because syphilis is a sexually transmitted infection. You know, so the diseases themselves are very different, but the connection here is that the bacteria that cause these two diseases are both called spirochetes, and spirochetes have axial filaments. Now, fimbria, which we had in the picture of the prokaryotic cell previously, fimbria are hair-like appendages that allow bacteria to attach to their host cell. And fimbria are significant because they are found on the bacteria that cause gonorrhea. So gonorrhea is another bacterial sexually transmitted infection. And if the fimbria are absent or removed in such a way, then the bacteria cannot cause the disease. So, you know, fimbria can be a target for things like antibiotics. And then pili, which is another thing that we saw in that picture of the bacterial cell. Pili are used by bacteria in a process called conjugation. Now, sometimes they're called sex pili because some, you know, people in some textbooks consider this to be a form of bacterial reproduction when it's really not reproduction. What they're doing is they are exchanging DNA with each other. And they're not really reproducing. They're not making more bacteria. It's basically an evolutionary strategy in which they are becoming stronger. They're getting more genetic diversity. So it may kind of sort of seem like a form of reproduction, but it's really not. But the term sex pili is still used very often. It's just not, you know, not the greatest term in the world because it's not really what they're doing, but still. It's okay. All right, so like I said, we definitely need to spend a good bit of time talking about the bacterial cell wall because the cell wall of prokaryotic cells is very unique, it's very special, it's very important for various different ways. So first of all, it's important because it's important to the bacteria. It provides them protection against things like lysis due to changes in osmotic pressure. That should hopefully sound somewhat familiar because you talked about things like hypertonic and hypotonic solutions in AMP1. The other thing that's very significant about the bacterial cell wall is that it can give some bacteria the ability to make us sick. So of course, that's important for the bacteria. It's also important for us to know about because that is the main focus of our class is, the bacteria that make us sick, the bad guys, the ones that can hurt us. We need to know everything we can about this bacteria because we need to fight it. We need to destroy it because we want to help our patients get better. So the main component of the bacterial cell wall is something called peptidoglycan. So you definitely want to know that. It's in the PowerPoint in bold, so you know that that is an important term. Now, peptidoglycan is a very complex macromolecule. It is made up of things like N-acetylglucosamine, N-acetylmuramic acid. It is linked by side chains and it has rows of polypeptides. It's a very complex molecule. And don't worry, you really don't need to know the details of that. You just need to know that the major component of the bacterial cell wall is peptidoglycan. But I do want to share with you just how complex this peptidoglycan is. And the main reason I'm sharing this with you is I just want you to feel my pain a little bit here because way back when I was in college, I was actually a pre-vet major. I did not become a veterinarian, which I think is probably for the best because I really like what I do now. But I took a class called Animal Diseases. And it was a class very similar to this class, except it was more focused on diseases that only affected other animals, not diseases of humans. But we took a test, and I remember very, very well. I turned the page of the test, and there was a single blank page. And at the top, it just said, draw the structure of the peptidoglycan molecule. And I about fell out of my seat. I could not believe that this teacher, who seemed like a very nice person, would actually ask us to have to draw this thing that you see on the left-hand side of your screen. Because I am not a good artist. I am not good at drawing stuff at all. And needless to say, I did not get very many points on that part of the test. But yes, so what you are looking at here on the left is essentially right here is the peptidoglycan structure. So this is what it's made up of, all these different proteins, all these different, polypeptide chains. And you'll notice that there are two different ones here. So peptidoglycan 1 and peptidoglycan 2. And you might be wondering, well, why are there two different pictures? There are two different pictures because there are two major types of bacteria, which is gram-positive bacteria and gram-negative bacteria. Now, this is really important because this is going to follow us throughout the entire semester. It's going to be important in lecture and in lab. It's very important to identifying bacteria, and it's just critical to know this information. So this actually comes from something that was created by someone whose last name was Graham. So gram should always be capitalized. It doesn't have anything to do with when you actually when you weigh something with a scale and you're weighing it in grams. Okay, so it doesn't have anything to do with mass or weight, nothing like that. The person's last name was Graham, and they invented this thing called Gram staining. Gram staining is a procedure that we are going to do in lab, and it is something that you do to a bacterial sample with different types of stains to figure out what type of bacteria it is. So if you, perform Gram staining and the sample stains purple, it is Gram positive. And if it stains pink, it is Gram negative. So you definitely want to know that Gram positive is purple and Gram negative is pink. Now they stain different colors because of the different structures of their peptidoglycan. So they're different cell walls. So Gram-positive bacteria have a thicker layer of peptidoglycan, and their peptidoglycan contains teichoic acid. Gram-negative bacteria have a thinner peptidoglycan, and theirs contains lipopolysaccharide, or LPS. So now if we go back to the picture here, you'll see that this peptidoglycan is much thicker and it has teichoic acid. This one is much thinner, but it has LPS. So this one is Gram positive and this one is Gram negative. That's why you have two different pictures, because you have two different types of bacteria based on the structure of their peptidoglycan. So I will definitely never ask you to draw the peptidoglycan. I won't ask you, the pictures and to identify the differences. Nothing crazy like that, all right? But you do want to know this information that's on this PowerPoint slide, okay? So you want to know the differences between Gram positive and Gram negative. So which one is purple, which one is pink, which one has the thicker layer, which one has the thinner layer, which one has teichoic acid, which one has LPS. Now, LPS is also called endotoxin. Endotoxin can make a person very sick. It can give us a very high fever. So in general, we do consider Gram-negative bacteria to be worse in terms of causing a worse infection, although there's plenty of Gram-positive bacteria that's really, really bad as well. But for the most part, Gram-positive bacteria is considered to be less dangerous, and it's also easier to kill with antibiotics like penicillin. So there you go. There are your two types of bacteria. So speaking of penicillin, we talked about, in chapter 1, we talked about Alexander Fleming and the discovery of penicillin and how absolutely critical that discovery was. And, it was probably one of the most significant discoveries, maybe of all time, maybe, in all of science. And, I would say probably it was the most significant discovery in medicine, in medical science. So the cell wall of bacteria is one of our key methods of killing the bacteria. So essentially, the cell wall is our target with antibiotics. Now, I know that the next video picks up with this, but I'm not exactly sure where I kind of pick up. It's just kind of weird, the video kind of cuts off in a little bit of a weird spot. So I just want to make sure that everything is covered, because this is a really important kind of discussion that I like to have. So in the, when the class is an in-person lecture, this is a slide that we spend a little bit more time on. And this is one where I do like to have a little bit of a discussion where I like to kind of, leave it up to the students to kind of think of a little bit. Now, of course, since this is being done, in a virtual kind of video type format, we can't really do that, and that's okay. So essentially, we target the cell wall with antibiotics. And that's the way that a lot of antibiotics work, is actually by disrupting or destroying the cell wall, because it destroys the peptidoglycan. But the reason why this is such a good thing is because peptidoglycan as a molecule is unique to the bacteria cell. What I mean by that is if something like penicillin targeted DNA or, a certain amino acid or a certain protein, okay, or something like a ribosome, all right, these are all things that our cells have in common with bacterial cells. So we would not want to be developing medicines and treatments to kill bacteria that focus on parts of the bacterial cell that our cells also contain, because then our cells would also get damaged, okay? Like we would not want to develop a medicine against bacteria that destroys DNA, because the DNA of bacteria is very similar to our DNA, and that medicine would destroy our DNA, which is not good. So the fact that things like penicillin destroy peptidoglycan, which is unique to bacteria, is fantastic, because that means that, antibiotics can focus on just destroying bacteria. It's also why antibiotics will, harm good bacteria. And that's why sometimes if you're on antibiotics for a long time, the doctors will put you on things called probiotics, or they'll encourage you to eat, more, fresh foods that contain, more good bacteria. Because, most antibiotics, they can't differentiate between good and bad bacteria. They just wipe out all the bacteria because they're destroying the peptidoglycan. So there you go. That's basically what we would discuss here. And I do believe the video picks up, or the next video picks up from here. So I'm going to go ahead and stop the recording.